So before I do go into, these are some wonderful people that are still in the lab. I had like maybe twice these numbers that have gone and taken other jobs, eventually become professors at other universities, or they're technicians. But these are the current ones that are helping the program both. So I have a large extension appointment as well. So we have a master beekeeper program. So you'll see a couple of them there that are helping with the master beekeeper. Without these people, I know I lead the honeybee lab, but I don't take credit for everything that happens from there, right? So maybe I'll take credit for everything good that happens. But without these wonderful people, I don't think I can run a program at OSU. So these are a few things I'll talk in the next. I have an hour, Norm? 1 hour? Okay. Yeah, I'm notorious. I can keep you until midnight as well, but I'll try to keep short. So we'll talk about European foul brood, then I'll go over an amitraz study that we did for the last few years. Some of you know. Has anyone not heard of amitraz before? Okay, the new beekeepers. So amitraz is a compound that we use to kill mites. I know Fonta was talking about mites and oxalic acid, so we'll go into oxalic a little bit today, but so amitraz is a very popular synthetic product. I know some of you hate using synthetics in your hives, that's fine, but you have to do something for mites, right? You can't just assume you have no mites or you think your mites are under control. They are not. So we'll go into one study at least with oxalic acid, then so honeybee nutrition is another important area that we focus at OSU. And if I say honeybee nutrition is an understudied area, don't get surprised. So when I did my PhD at Texas A&M, there was a guy who used to work with grasshoppers, and they knew more about grasshopper nutrition when compared to honeybees, what we know today. So it's not like honeybees are not important or anything. Honeybees have been studied for more than 200 years in the modern world with a very designed studies, but still we have barely scratched the surface. We only know basics, right? You all know bees need nectar or honey for their carbohydrate source. We know pollen is their protein, and we know several vitamins and other things that we need. But there are so many micronutrients that bees need, like you all probably take some vitamin tablets or other things, right, in your daily life. But with bees, we don't even know what are the sterol requirements. Sterols, like you have cholesterol, they have something called 24-methylene cholesterol. So we don't know any of those requirements and how much we need. So our basic program with honeybee nutrition focuses on understanding all those needs, and then we do applied studies with protein supplements. People feed protein supplements all the time, right? So we want to see, are they useful or are you wasting your money? So I work very closely with commercial beekeepers as well, and so their bees, at least for some of you, it's a backyard operation or you are a hobby beekeeper, and I know we all get sad when you lose bees, but for a commercial beekeeper, it's a living, right? So they have 5,000-6,000 hives, and if they lose half of them every year, they are in trouble. So we do work closely with them on several projects, including looking at protein supplements they feed and to see if they have any value. So I'm going to show 1 or 2 studies on that front as well. And then the last one, just before I planned on coming here this evening, I put some slides for tropilelaps. Have you heard of tropilelaps? Some of you, know, the experts here, Fanta or Norm or Pam or Nancy, they might have heard, or Matt. But what about the new people or those who have kept bees last 2 or 3 years? Have you heard of this tropilelaps? Okay, what is that? Those who have heard of it. Yeah, no.
Isn't that out of Southeast Asia?
Yeah, so it's a mite. So it's tropilelaps is a mite. As Norm said, it's mostly in Southeast Asia. Are you guys worried about this? It's not in the US yet. So I talked about varroa, right? Varroa is there in the United States, has been there for more than 30 years. But tropilelaps is not there in the United States, and there are people worried about it, but is anyone here worried about tropilelaps? Oh, there are a couple of hands. Some of you are not worried. Why do you think you are not worried? Weather, okay, that's one good point. What else? Or do you think it's a very big name, maybe it doesn't matter, it doesn't come here in the United States? So, yeah.
I am worried.
Yeah, right. So yeah, I mean, I will not scare you, but you probably heard a lot, right? Recently, there is Auburn University, there is a researcher, Geoff Williams, he's doing a lot of work on, so it's not here. So if you want to study this mite, you have to go to either either Thailand or South Korea or China or India or Bangladesh or some country in the Southeast Asia. You have to go to study this. You can't study this here in the United States. But I have a vial. I didn't realize the lighting is not that bright here, but I got a vial where we have couple of tropile labs if you want to see. You can see with your naked eye, but you need a very good eyesight for that. They're very small. Your varroa is a lot bigger. They are only 1/3 the size of a varroa mite. If you have seen a varroa mite, they are either 1/3 or even smaller in some cases, depending on the species. So I have a vial of those. Don't worry, I didn't bring any live ones here to spread here in the United States. These are samples. We got dead samples in alcohol for those of you who are curious to see. And I mean, we should monitor. I'm not going to scare you. I know some people are scaring already, saying, oh, this mite is going to come. We did talk about this mite probably 20 years ago as well. It's nothing new. The Southeast Asian countries, they had this mite for hundreds of years now. But, or at least in one species, Apis dorsata. Then they spread into Apis mellifera, the bee that we keep, the European honeybee. So yeah, again, I'll not waste time here. When those slides come, I'll give you more biology. But then, yeah, be prepared for learning a little bit about this new mite, which is not here in the United States, but people are thinking maybe in the next 10, 15, 20 years, it might arrive accidentally because we do a lot of shipments, right? Like Canada imports bees from Ukraine, and we think Ukraine already has that mite. So it might come through Canada to US or maybe to some other countries, if not directly into the United States. So we'll talk a little bit about that as well today. Okay, let me start with this first one, European foul brood study. And before I wanted to thank you. I know you always invite me in January. I thought maybe I'm very important. That's why you guys invite me in. I was joking with Pam. Is that the reason, or you think January is winter, no one else will come, so this guy will show up from Corvallis? Okay. I'm just joking. No, I really enjoy coming to your group. I have seen this group for all 15 years now plus, so I really appreciate that you guys are really interested in this topic and very knowledgeable. I get a lot of great questions from your group as well. So European foul brood, has anyone not heard about this disease before? Don't be shy, you may be new, so raise your hands and then I can go into more details. Otherwise, I'll assume that you all know what European foul brood disease, and I'll directly skip to tropile then. Okay. So this is a brood disease. When I say brood for the new beekeepers, just imagine brood is mostly the larval and pupal stages we use that term for. So it's a brood disease, right? So you can see that's a long name. We don't need to worry about it. Melissococcus plutonius, that's the organism that causes. It's a bacterial disease. So most of the larvae that you see are killed is that stage, 4 to 5 days old. And then the sick larvae, they first become displaced in their cells. I'll show you some good pictures in a minute. And then the larvae turn flaccid, like more fluffy, right? Not fluffy, I think liquidy. And then you can see they turn brown and they die. So you're seeing some healthy larvae on your left, and you see some of the European foul brood disease larvae. You see they are turning into brown and they are melting and dying inside, right? So I'll show you a good image that you can really think of. So you see here, so I said they get displaced in the cell, right? The larvae are displaced. Who is displacing them? Can larvae move inside a cell? How many of you think larvae can move inside a cell? Only few of you? Okay. And the others, you think they are just sitting there and not moving at all? Yeah, because when you open a frame and look at those larvae, right, they are not moving, right? They are not moving and trying to figure out where the food is, because it's a progressive feeding. There are nurse bees that are coming and feeding the larvae. They directly put that brood food close to their mouth parts and they are eating. But if you starve the larva, if they are not getting the food, they will twist and move slightly. But you have to continuously record it to see the moment. You can't just go and say, oh, larva, is it moving or not? So for those who have never seen a larva moving, I'll show you a video here so that you believe me that larva can move. It's a slow video. Just watch some of those larvae. A couple of them you will see they are moving. Did you see the one in the middle? It's trying to crawl out of her cell. The reason being, you can do this experiment at home. It's not an experiment technically. So bring a frame. If you have your hives just close to your house, bring a frame just for fun that has 4-day, 5-day-old larvae and put near the sink and allow them for an hour or two. And you will see some of them crawling out and falling into your sink. Because there is no one feeding them, they are looking for food. So they are just wandering around and they fall. So this is something I'm showing you. We brought something to the lab and that's what you're seeing, a larva crawling out. So those are honeybee larva, right? So larvae do, so again, just for fun I'm showing you, but the whole reason why I'm showing you is to show that the larva will get displaced inside the cells like that if they are looking for food. So this disease is mostly related to nutrition as well. We think, again, we are testing several hypotheses. We think poor nutrition could be one reason why you see this flare-up of this disease. The bacterial inoculum is already there, but when the disease starts, when there is a problem with stress, it could be nutritional stress, it could be others. We'll talk about those in a minute. Any questions? Okay. So I'll just show you one where I think Carolyn took this video a few years ago. I'll see if that works.
Here is a classic example of European foul brood. First we note the spotty brood pattern, and then at a closer look we can see larvae in the cells, in open cells that are discolored, yellowish, and melting towards the bottom of the cell. To make sure it's not American foul brood, we'll do a ropey test. So we have a stick, a wooden stick. We'll take it and stir around a larva inside of a capped cell, pull it out, and see if it ropes. It's not roping to look like an American foul brood. We'll do one more. Capped cell, stir it up, and pull it out, no ropiness. So it's not American foul brood. Here is a classic example.
So you probably heard of ropiness test, right? I know your group, I think Fonda or someone, Norm or someone will tell you when you get into more bee biology and the bee school concept. So American foul brood is the nasty disease. That's what you don't want in your hives. And accidentally, if you happen to have, I think get rid of those hives. Because I have seen a lot of problems here. I'll just give you a quick example how bad this disease can be. So this was like 7 or 8 years ago. There was a truck crashed on 97 on Madras, Central Oregon, if you know where that is. A beekeeper was going from California almonds pollination and going back to Washington. And the truck was in a crash and the bees were all over the place on the road. And then I got a call from ODOT and other people asking, do you want to rescue these bees? Otherwise, we are going to burn all these bees. They just wanted to put gasoline and burn because they were being a hazard for people that are driving around those trucks. And what a waste of, sheer waste of bees, right? And we don't want to kill that many bees like that. So I called the Bend Beekeepers Association and I said, go look at those hives, don't take them home, put them in an isolated area for at least one brood season. And then if you don't see any disease, then take those back home and distribute. These were free hives, right? Because the beekeeper had an insurance and he said, I don't want those bees. So no one was claiming. So the ODOT just wanted to burn those hives because it was a hazard. So they didn't listen. Some of them, they took the hives directly home. And then next year on, I used to get these calls from them that they are seeing American foul brood disease. But Bend area didn't have the disease that much before. So that's what I'm saying. It's a very nasty disease. It's not this one. The one I'm showing you, this is more milder. Even if you have this, you will get rid of this at some point. But American foul brood, you saw that Carolyn was taking the stick and pulling. If you get a string, probably you have an American foul brood. But then you have to use the testing kits we have, or you have to send it to our lab or to Beltsville to do a positive identification. You don't want to burn your eyes without confirming that that's the American foul brood disease. But this is a different disease I'm talking. This is also a bacterial disease, but it's called European foul brood. This does not produce spores. But American foul brood produces spores. Those spores can be viable for 60, 70, 80 years as well, some studies have shown. So you don't want that disease. I hope none of you have that, but in case if you have, then please burn those hives. There's no other option for those. Okay, so yeah, it could be a problem in some areas if you're not careful. So again, I know I already wasted a few minutes here, so I'll be quick on this. I use the term enigmatic disease. Enigmatic disease means it's mysterious, right? It has been studied for probably 70, 80 years now, very carefully, but still we don't know what are the triggers for this disease. Why do we see this in spring and then sometimes it goes away and then comes back? So that's why I'm using this term enigmatic disease. So it's a seasonal outbreaks you see. I probably have cited some old studies because EFB was not studied well after 80s. I think people just ignored it a little bit. Now we are seeing more of this in commercial beekeepers that do blueberry pollination. So we are focusing on this disease. So we got a large USDA grant year and a half ago, so I'll go into that study in a second. So probably you all know, right? EFB is a cyclical disease, like people say. In spring I see, and then it goes away, and I never see the disease until next year. That's probably many beekeepers have that experience. And I'll show you a graph why that is. So I know it's not a great, it's an old book that I had to take a picture of, so I apologize if you can't see very well. But what you're seeing that, you see those dark dots that are going up. So what you have is disease larva on that y-axis, and the x-axis is months, right? So June, July is the time in many places the nectar flow is pretty good, right? So we see that during the main nectar flow, the disease goes up and then comes down again, right? You don't see much by end of July or August. So what these people were speculating at that time is, so what happens, think about what happens during nectar foraging. There are too many bees that are recruited to do foraging, right? They go out to get nectar. So I'll give you one more here. This is an example. So I made a graphic for you to understand. So you see there are 30,000 bees. I'm just making up some numbers for the new beekeepers here. So we have about 30,000 bees. And so if I pull a frame, there are about 3,000 larvae. Again, I'm just making up some numbers. And there are 6,000 nurse bees that are attending those larvae, right? That's an example. Nectar flow started. You still have that many bees in the hive. But what happened is they have recruited more bees. Sometimes you can see precocious foraging. The term we use, precocious, means they are foraging before their turn comes. Usually the bees will forage, you probably learned in your bee biology, they should be at least 21 days old when they start foraging, right? But in some studies people have shown they can forage at 15 days old as well, depending on their physiology, the environmental conditions, a lot of different things. So precocious foraging is not uncommon. You can recruit bees earlier than what they are supposed to go out. So in this case, when their nectar foraging is happening, there is a lot of stimulation to get nectar. There are a lot of bees coming and dancing and recruiting other bees. So what could happen is there will be more bees that are going for foraging, and you lose some of those nurse bees, right? Some of the nurse bees might have started foraging as well. So what I'm saying here, just look at the right side here, there are 3,000 larvae. The larvae are not going anywhere, right? They're not going to fly out from the hive. They're still there in their frames. But instead of 6,000 nurse bees, you have only 3,000 now, because many of them started foraging. So what happens? There were 6,000, there is 1:2 ratio, right? Almost there. So there were one larva is fed by 2 bees, and now you got only 1 bee here for 1 larva. So isn't that a compromised nutrition? Because they are not being fed that much now. So that would compromise their nutrition. And then the inoculum, the bacteria is already there. If you had the disease, it's there on the frames, on the comb as well. It's just waiting for perfect conditions. So when you stress them by doing something like this, then that disease shows up. The larval immune system is compromised because the nutrition has been compromised, and you get the disease. So that's a basic hypothesis. That's again one. I'm not saying this is the only way that they're getting the disease, but this could be one way how they get the disease as well. Is it clear, or did I confuse you? Okay. So why EFB now, and why we are studying this? At least several commercial beekeepers that do blueberry pollination have been complaining, not just in Oregon, Washington. So blueberries are not grown everywhere, right? There is some acreage in California. Oregon is probably 2nd or 3rd largest producer. There is Michigan that produces blueberries, Washington. So all these states, beekeepers, and then British Columbia, of course. There are a couple of beekeepers that do most of the pollination in British Columbia. So they have all been complaining about this EFB disease for the last 5 or 6 years, and no one really knows what's happening. And some of them have stopped going to blueberry pollination because they lose their hives. Because EFB will not totally kill your hive, but it will weaken your hive that they're not good pollinating units. And maybe eventually, if nothing happens, they may die as well. So that's why we wanted to look into this problem. Why blueberry, right? So here I'm just showing you a beekeeper from British Columbia. It was a big news in British Columbia. So here it says beekeepers refused to put colonies in Fraser Valley blueberry fields. They just didn't want to because of EFB problems. So here are a few hypotheses here that we are testing, looking at nutritional stress, fungicide exposure, because crops are sprayed for controlling fungus or fungal diseases. So when you are in blueberries, there will be some fungicides that will be applied. Brood chilling, I'll show you a video. You all know, living in Oregon for this long in the valley, you know how the weather is in April and May when blueberries are in bloom. So it could result in brood chilling. I'll show you a video of that too. And then we are speculating there might be a new variant, right? You probably are all tired of COVID. You heard from what, Omicron to Delta to whatever the strains, so many strains. So virus mutates, right? So bacteria can have mutations as well. So we are suspecting there might be a new variant of EFB because no one has studied EFB for a while. So we think there might be a new variant of EFB that could be also causing this disease. It's not going away. It should go away after a month after nectar flow, but it's not. So we see this disease even in August and September as well, even in almonds. So we are going for this study February 15th. I'll be in California looking at some of the hives that commercial beekeepers have for the experiment, and I expect to see the disease there as well. So it's not going away. So we want to see, is it a new variant? And of course there is a hypothesis which I really don't give much value to it. I think some people think the blueberry pollen has a little more acidic pH, and that might create more problems. But then the reason why I don't take this hypothesis seriously is bees don't get much pollen from blueberries. If you have done any blueberry pollination or heard of it, blueberries are a painful flower. Your honeybees don't even like getting in there. But most of the nectar foragers, they are the ones doing pollination. You will not see too many bees bringing pollen from blueberries. And we have tested with pollen traps, and we have analyzed pollen with microscopic and other methods. It's only 4% of the pollen that comes from blueberries. They are going to some Scotch broom or viburnum or some other weeds when blueberries are in bloom. But they do pollination of blueberries. Don't get me wrong. Or a farmer may say, "Am I just foolish to rent bees and if they're not collecting any pollen?" So you should tell them, the nectar foragers are doing the pollination, not the pollen foragers. For new beekeepers here, the foragers are different as well, right? They partition their work into pollen foragers and nectar, and some will get both as well. There are some bees that will get both pollen and nectar, but most of the time they will be either collecting nectar or pollen. So the nectar foragers, they do go into the flowers, and when they go in, there are 10 or 15 pollen grains. They end up on their antenna, mouth parts, forelegs, and that's enough to do pollination. So most of your nectar foragers do the pollination in blueberries, not the pollen foragers. So the other hypothesis we have is splits, right? When commercial beekeepers, probably you will learn about splitting as well in your group at some point. When your hives go too strong, you have to split them, right? And people have their own methods of splitting hives. The commercial beekeepers, what they do, so suppose you have 100 hives in Oregon, you take 50 of them to California and bring them back, they will be twice the size of the ones you left home, because they get so much pollen in almonds, the colonies just grow crazy in that 1.5 month or 1 month, whatever time they are in. So those beekeepers, when they come back, the commercial beekeepers, they make splits to either sell to you guys as backyard beekeepers or to some other people, or just to increase their numbers, they do splits, right? So splitting could be a problem as well. That could be adding to the disease. Can you think about how that can be? Yeah, Wanda.
I was going to say, I'm so glad you brought this up, because I think this is the application where we're going to see it in our hives, is that I've had it happen a couple times, and I've seen it in a lot of your guys' hives too, where you make a split when the temperatures are great and the bees build up. You make that split, and either all the bees go back home, and the one that you've made the split, right, has got less foragers, and then they become stressed, and then they pop out with EFB.
Yeah.
And then the drones spread it between the hives.
Yeah, great point. Yeah.
Or you buy a nuke or a package, and you don't feed it enough sugar in the beginning to get it started right. And then, because the population is dwindling, it stresses them out, they get EFB, and then they spread it around the apiary.
Yeah. So what Fonda is saying is you have skewed up the demography in the colony, right? You have skewed the population dynamics in the hive. So as she said, foragers flew out, now you have skewed the population. So that's what we are looking at. So the spreading could cause a nutritional stress as well. I don't know if I have an example to show now. So what happens is, if you are stealing some nurse bees from here, or maybe, like as she said, all the foragers moved out, only nurse bees left, or maybe with commercial beekeepers, what happens is they will just randomly pick sometimes frames of bees from here and just put this in empty box, and so that random removal of frames will skew the population dynamics. That's what I mean. So if you have less number of nurse bees, for example, I had 2 hives, and this is the one I'm splitting. This is the one I'm splitting, and I have an empty box here. I bought a new queen, and now I am putting all these frames from here and there. I steal 2 from there, 3 from there. If I take too many nurse bees from here, and there is still larva enough, then I have screwed up, right? Because I showed you that there are more mouths to feed than the nurse bees that are present. So that would create a problem. So that's what we think splitting also, like as Fanda said, in a different scenario, but in commercial beekeepers, I think that's what is happening, is sometimes they are not very careful in systematically taking enough nurse bees, enough foragers, and it's hard as well. And there is no practical way I can tell them that, but we are testing this hypothesis. So we will do a very focused experiment next year where we will steal frames of bees from several hives. We have like 40 or 50 hives that we'll use for experiment, and then we'll spray them with the European foul brood spore, the inoculum, and then we'll see the bees that have been skewed in terms of population, maybe they get the disease and not the ones that already have a good dynamics of population. So again, this is another thing we are going to test as well. This is the weather I was talking. I know you all live in Oregon, so I don't have to show you this, but this is the crappy weather you see, right? So the blueberries are in the background there, and you see those hives are being rained on. So the bees don't have access for almost a week sometime to forage, or maybe at least 2 or 3 days sometimes, right? So that could result in brood chilling as well because it's cold, and then bees are not flying. So there is a nutritional stress. Again, nutritional stress is not just because you're screwing up their population dynamics. It could be because of this as well, because bees are not foraging for a week. So they have eaten up all the stores that were there already, because they don't store too much pollen like nectar, right? Or nectar or honey. So there is only, there are not too many studies, but at least one study from Tom Seeley or someone had shown before, they don't store like more than a kilogram of pollen in the hive at any point of time. That's just a rainy day thing. So when you are in rain for a week, they eat up everything that is there. So that could result in nutritional stress as well. So we did some studies in the past. This is one publication. Ellen, one of my students, probably this is 6 years old. I don't know, is there a date? Yeah, 2019, 5 years old probably. So we looked at different crops in the West Coast here to see how much pollen from each of those crops comes in. So you can see here, that's blueberries there. So it's only 81 grams. I know science is all in grams and kilograms, so I apologize. You may have to convert into your ounces or pounds. Do you use your calculator later? So blueberries, you see, is one of the lowest ones. Hybrid carrot seed is still worse in Madras, but you see only 81 grams per colony per week. That's a very small amount. So that causes a lot of stress on your bees when you are in crops such as blueberries. So this is a huge grant we got with several collaborators. Have some from OSU that you're seeing on the top, including Anthony, one of my colleagues. And then we have on the bottom, there are UC Davis, WSU, and Mississippi State as well involved. So we started this in last year in February. We are in California there. You see all teams from different states there. These are some objectives. We are looking at the prevalence and intensity of the disease. We'll look at some techniques to manage EFB. And we also look at any genetic variation, because I talked about those new variants that might happen. And then there are a couple of other objectives. I didn't put everything here. So basically, just to be on time, I'm not going to go in detail here. But how much time did I already kill, Norm? Oh, I'm already gone 30 minutes. Okay. So I should be going faster, or else I'll not get to trophy lapse. Okay, so on the bottom, what you're saying is the poor larval nutrition. It could be because of skewed larvae. I talked about that. Optimal foraging because there is bad weather. I showed you that video of rain. And then some landscapes you may not have enough pollen, even if you are in a good weather, but everything around you doesn't produce much pollen. That could also result in poor larval nutrition. And those are the different hypotheses I said. Inclement weather, brood chill, the bees are in blueberries. So that's how we start in almonds, then we go to blueberries. So we are looking at all these different hypotheses. I think the next slide has the design here. So that's what we have been doing, 1536. It's one of the largest studies in the United States looking at EFB. No one has done this large scale study. It's a pain to, again, have that many collaborators, beekeepers. I know if you have worked with a commercial beekeeper, you'll understand they're very generous in allowing us to do whatever to those hives. but then just to ask them, it's a big deal, right? We ask them to put in different crops or whatever they are doing. So it's a huge endeavor to really deal with. Sometimes the beekeeper probably tells his employees or her employees to put those bees. Sometimes they forget in where they have. So a lot of management goes on. So again, long story short, half of those colonies that go to almonds, they went to blueberries, like 768, and then 768 went to another crop or holding yard or wherever. And then they all came back to 3 more different locations. And then, so this is a 3-4 year study, but we are repeating this twice. So last year, so those all hives have tags with OSU numbers on them, and we follow those same ones, right? You can't just randomly use another hive next year, because you need to have the history of EFB for those hives. So same hives, and we selected that many because beekeepers lose hives in winter. So we are expecting maybe 30-40% loss. Then also we'll still have enough hives this year to do the work. So that's our design that we have for that. So again, I'll give you more updates next year when we talk. It just started this year, so I don't have all the data. I have some preliminary data here for you. So these are the 4 beekeepers, Stephen Kaufman, Tom Cinquini, Karen Finley, close to our place in Philomath, and then George Hansen in Colton. So this was our first sampling event. We do a lot of colony evaluations. Each hive takes 30 minutes. So imagine 1536 it can kill. When we got this money, I was excited, but now I go with my crew. I think Paula, you came for one of those training things, right? So you saw how much work it is to really, it takes several days just to finish one beekeeper sometimes. So we are doing a lot of colony evaluations, looking at EFB scores, then brood area, bee populations. When we see a disease, we do use these kits. Has anyone used these kits before? Fonta, have you used? No? So, just one time? Okay. They're expensive, about $14 or something, but I think they're reliable. I would say 80% or 89%. Sorry, there was a study that said they are accurate 89% of the time. I think that's the best you can get. I don't think you can get 100% anytime with any of those type of kits. So we do PCR as well. This is just a field confirmation, but then all those larvae we collect, they go for a PCR diagnostics as well at OSU. So here are some larvae that we are collecting. You're seeing, this is just a joke. If none of these techniques work, then you can use your dog to detect. So it's not a joke. Actually, there are a couple of apiary inspection programs in the country that they have trained their dogs to sniff the disease. Not this one, American foul brood. Have you heard of that before? Yeah. So there is, I think University of Maryland has one dog that is trained to detect American foul brood, and then there is one more state somewhere. This is my dog. My dog doesn't know anything, so I just. So you, yeah, sorry. So we collect nurse bees as well, because we want to make this correlation, right? So if I get a score of EFB score, so we have 1 to 10 larvae, we give them a score of 1. If you find 11 to 50, anywhere you get a score of 2. If beyond 50, it gets a score of 3. So we give them scoring, and so the whole reason we collect nurse bees as well from the brood area, assuming that most of them are nurse bees. Then we do vitellogenin and protein analysis, so that you can have a nutritional hypothesis tested, right? If I see a score of 1 and I had a nutritional status because of those nurse bees, again I'll just give some maybe 3 nanograms protein, and then when the score goes to 3, that means the disease has increased. Is this because the nanogram of protein has decreased from 3 to 1 or so? So we can make those correlations as well. So it's a very robust study where we are trying to make correlations between all this head protein, vitellogenin, to the disease scores as well. We do a lot of pollen trapping to look for fungicides and any insecticides that are in the pollen in those cropping systems wherever those bees are. Here you can see some of those. Again, this is just a pattern I'm showing you. So that's beekeeper 1, 2, 3, 4. Just from the Oregon ones you can see. So you see the ones that are in non-blueberries, the orange ones, and then the blueberry ones are the blue ones. So except for this number 3, we still are trying to understand what's going on. The others, all the disease increased in the intensity when they were in blueberries. And this is a cool vitellogenin analysis. So we can see the gene expression for The ones with EFB 0, we expect them to have good protein, right? So vitellogenin is a glycolipoprotein, really important for your bees. You can see that EFB score 3. So we randomly selected some from, so it's a large study, right? We have thousands of samples of larvae. So I just asked them to get some preliminary analysis. So we are seeing some good trends. You see the EFB score 3 is the lowest one. So it looks like our hypothesis might work if we get all the data analyzed. We'll see. Okay, I'll switch gears here because I'm running out of time. So we did some amitraz testing. For those of you who don't know, this is a synthetic product that has been used in the United States for probably 15 years plus now. And how many of you have used synthetic product like this before? Just 1, 2, 3, okay. What do you guys think? It works? Oh, here is the main guy here. I didn't see him, so I'll be in trouble if I say something wrong here. Have you used? Right, yeah. So what do you think? APOR works or it doesn't work? APOR, the product.
Yeah, I've stopped using it.
Okay. So some of you have stopped using it. And what about the other hands that I saw? You still think APOR works, the amitraz product?
No, we went to oxalic acid.
Yeah, oxalic is good. I know you should use oxalic, but I'm just asking about this, yeah.
We tried it one time.
Okay.
At the end of the treatment, our mites had gone up.
Gone up, okay. And what time of the year was it?
Oh, it was late summer.
Late summer, yeah. So there is a problem with this. I'm not saying it's all working. So I'll show you some data quickly and summarize what we are finding. So amitraz is a good product. It was probably one of the best ones because we didn't see much resistance until now. We are seeing some pockets of resistance here and there. I'll talk about that in a second. So this is a very popular product, and that's what I say. If you lose this tool, then you don't have enough synthetic products. I know some of you don't like using synthetic products, but I think commercial beekeepers, they don't have a choice. They have to use something to kill those mites, right? So this has been a very important product. It's expensive as well. It's not cheap. Those strips are not that cheap, right? But then commercial beekeepers have used a product with a different formulation, which is not registered for use in the hives. But they use, and again, I don't blame them for it because as a researcher, I know I should be sympathetic for any of those causes. And so they use it's for cattle ticks. So same amitraz is sold in a liquid formulation like emulsifiable concentrate EC to kill ticks in cattle. So some beekeepers will use those and put them in shop towels and put in the hives, which is technically illegal, right? I hope there is no ODA person here or Oregon Department of Ag in the audience. Even if they are, they should be sympathetic to beekeepers because beekeepers don't have much choice, especially commercial beekeepers. We haven't provided them enough tools to control mites. So oxalic and all are great, but commercial beekeepers don't have time to use oxalic all the time, right? As you can. So again, long story short, so we wanted to see, is there resistance problem, and if it is, then what to do with that. So that's the study, right? And this is all done in Madras because it's a nice location. All the carrot seed production is there, and we get about, I think in the best year when there is a lot of acreage, maybe 15,000 hives, or else it will be less, 10, 8, whatever. But you get a mix of beekeepers from Oregon. There will be a couple of them from Washington, couple from California. So we wanted to use that location so that I'm not running around finding each beekeeper every day, where are you, and then going sampling. So we just went as a big crew and spent 2 days. I think Matt came as well once or twice from your group as a master beekeeper. And yeah, we did this study. So I'll just quickly go into some results. And so this was in 2019. So you're seeing some of my crew doing some of those testing for, and we were trying to compare Canadian populations as well, because Canada has never used that illegal formulation. So we thought maybe we'll see a big difference between Canada and USA, USA meaning just Oregon, right? So at least we are, so in this year we saw some significant difference, right? You're seeing an 86 and 97, but then when we repeated the study in 2019, we didn't see a significant difference there. They are almost statistically similar in numbers. So we are still getting 91% control in US, and they are getting about 92.5. So that's a very broad study we did, but then I'll show you the recent study that we did. But this is what I want you to look at. So these are the letters you see on your left, A, B, C, D. These are beekeepers, because we keep everything anonymous. We don't tell who the beekeeper is, but you can see in the middle, you see all those good numbers, right? They are either in 80s or 90s, which is still a decent control for amitraz. But then on the right side, on the extreme right, what you're seeing, these are the outliers. For example, look at the top one, A, that beekeeper got 91% control, but then one of the hives got only 50% control. That's not a good one. It's a disaster. If you're getting only 50 or even less, that's not good. That tells me that there is some issue with the product or with mite populations getting resistance to this emitraz. So that's the reason we are still worrying and trying to understand. I don't have a good answer yet, but we are going to repeat that study again in Madras this year as well. So this was in 2023 and 24. That's what you see Matt in one of those top images somewhere. So this is a different cage design. There is a guy at USDA, Frank Rinkovich. He has done more of these studies. So we are following the same cage design. You see those cups. There are bees hanging on the top onto that APR strip, which is 4 by 4 centimeters, and then they stay in there for 24 hours, and we take drops. So you see all those yellow sticky cards, those mites will fall off from the bees once they are exposed to that miticide, right? So that's how you do the study for that. So I'll skip his study here, Rinkovich. These are our results. You can see in 3 hours we get about 36, 57, 86, right? So, but according to him, in Florida, he thinks all the mites should be done in 3 hours. So he's getting, so his mite populations are susceptible. He claims they have not used emitraz for 15 or 20 years. They have a bee genetics called pole line. I don't know if you've heard of those, but they have some mites. So I'm still arguing with him like, no, 3 hours, if I tell and show you guys and say, see, in 3 hours all mites should die, but there are only 36% dead, then you are probably going to kill me and say, oh, that means we have resistance, we should stop using this product from tomorrow. But no, don't get me wrong. Look at this 24-hour data. I think this is more reliable. This is what your degree of resistance probably. Again, these are efficacy tests. I should be very careful when I use the term efficacy versus resistance. Do you understand when I say resistance and efficacy, it's all same? No, efficacy testing is done to figure out resistance. If I'm getting poor efficacy, then I'll say, yeah, there is a problem with resistance, and resistance tests are done in the lab, not like just doing with alcohol washes or shakes. So that's a different story. You have to do something like this. So we started doing something called a vial bioassay. So you coat each of those vials with a certain amount of concentration of emitraz, and you put 10 mites with the pupa in there. I don't know if you can see here, there is a pupa and there are mites inside those vials. So these are more focused studies. So we just started doing this last year. So hopefully we'll have more data, and then I'll show you whether we really have resistance or not. 15 minutes, see, Norm is kicking me out already. Just joking, I'm notorious. So I'll be fast, I know. But I thought will have enough time, so I put some fun videos here as well. So you don't want to watch this one?
Yes.
Don't want to or want to?
Yes.
Okay. So have you heard of queens fighting together? Maybe in Fonda or someone else who have already talked about queen dueling. How many of you don't know what queen dueling is? Oh, okay, the new beekeepers here. So the queens do fight, not for fun, but when swarming season, like May or June, they produce a lot of queens, maybe a dozen sometimes. And then the old queen with half of the bees swarms out, right? And because they have already the insurance inside that there will be at least a dozen queens that will hatch at some point. So if there are 2 queens they hatch at the same time, then they try to find each other and then they will fight. Because usually you end up with only 1 queen. Sometimes they may both get injured and there will be no queen as well. So that's possible as well, but probably 90% of the time one survives. So this is what I'm showing you, but I want you to look at something called spraying behavior. I know I probably have shown this video to some of you, might have seen this video, right? Pam, you have not seen? You come to OSBA conferences. Have you not seen the video before? Okay. I have played this before. That's good. If you have never seen this video, then I'm in good shape here. So watch for that spraying behavior. This is very unique. So you guys know Randy Oliver, probably you read some of his articles and stuff. So he was in the audience when I was speaking at the Washington State Beekeepers Conference, and usually Randy is very knowledgeable. He knows most of the stuff. He has been into bees for 30 plus years or whatever. He was sitting in the front, and I asked people, what is the spraying behavior? Have you heard of this? And Randy also raised his hand that he never heard. I was, oh, Randy, I stumped you. You also don't know spraying behavior. it's something very unique. Not from many people have documented this, and there are only a couple of papers. If you look for David Tarpey, he's one of my colleagues in North Carolina. Tarpey is his last name, Tarpy, and look for spraying behavior. There are a couple of papers he has published on in 2004 or 5, sometime around. So spraying behavior, I'll show you the video and then we'll talk about spraying behavior. So these are 2 queens that we put on the lab bench to fight. And most of you think only the virgins fight together, right? Is that what the concept is? How many of you think only the virgin queens that come out of their cells only fight? Can't the mated queens fight? How many of you think mated queens can fight as well? Oh, okay, Norm is already raising. I see Dick there, and Dick, have you seen mated queens fight? No? Okay. So yeah, mated queens can fight as well. These are 2 mated queens. They are just lying on a lab bench desk and they are fighting. So when you get 2 different queens not related, they will come and fight. Watch the Dark Queen. Keep watching the Dark Queen. Something will come. Did you see something coming out? What is that? What was that? Spraying, yeah. What did she spray? Pheromone, yeah. Getting close, yeah. Okay. Yeah, I'm going to trip on this today, yeah. So, not many people know about this. I think the only 2 papers I mentioned, right, they have characterized the compound. It has some esters and other things in there, but yeah, something close to pheromone you can think about. That's a good analogy for sure. So their hypothesis is this queen was supposed to spray that on the other queen, but in this case she was not able to spray exactly because it's not in the hive. They are messed up on the lab bench. So the whole target was the other queen. So apparently this compound that she sprayed is so attractive to the workers. If the other queen that was injured more, the lighter one, then the workers will come and ball her, and immobilize her so that she doesn't move, and they may either suffocate her and kill. Again, this is all hypothesis again, but it makes sense. So that is very attractive to bees, the thing that was sprayed on, and that's what we know at this point. But please read the papers or email me, I'll send you the papers to read. So that's about spraying behavior. So if not anything, you learned something new today, right? Which is not very common. Again, I'm joking. And I'll show you this video, and then I have 5-7 minutes I'll take to wind up. Washboarding behavior. Probably this you all know, right? What is washboarding? Just this? Okay. Doing this? Yeah, that's what you're seeing there. So I'll show you a video and then we'll talk a little bit about that. So they're doing this rocking thing, right? Back and forth. Mm-hmm. So some people think they're cleaning the hive. I don't think there is anything cleaning there, right? And then some people think they are probably releasing some pheromone there to orient. That's not an orientation pheromone. You probably heard from your group, Nasanov pheromone that comes from their Turgel gland here on the back, but they're not facing back or something. They're just on the front going back and forth. So for the new beekeepers here, it's fascinating. You may see in your hives at some point. It's not very common either. It happens in the afternoons when it's very warm, probably between 1 and 4. People have seen most of this behavior, but no one really knows what they are doing. So it's still a mystery. So we call it a washboarding behavior because they are just cleaning something, people think. So you can still win a Nobel Prize if you figure out what they're doing, which we haven't figured out yet. So it's called washboarding, but that's what we know. And there are papers on this. So only thing that is really known well, because people have painted some different age groups of bees, and so the only thing that is known about washboarding behavior is those bees that are doing the behavior are middle-aged bees. So I would say middle age for a bee would be anywhere between 12 days to 18 days. That's the age group that is doing that thing. So, okay. Oxalic, I know this is a really important topic for you, so I'll just spend a few more minutes on this. So we have been doing some oxalic studies for last 3 years now. We just submitted a manuscript, so hopefully that will come out in the next couple of months. If it gets accepted, we'll see. So we have been testing. You all know that 1 gram per brood chamber doesn't work. That's not a good dose. So that's one thing I'm going to tell you before I even show you any of the data. And then we started looking at 3 grams and 4 grams because we thought that's where we were getting good control. But then you want it to be safe to your larva as well, right? You don't want to kill a bunch of larvae by using a high concentration. So we tested 3 and 4. So those are the results I'm going to show you. So this is the experimental design, 3 grams, 4 grams per brood chamber. Again, for new beekeepers, remember, you have 2 boxes. So for 1 box, I'm talking about that dose. 3 grams means for 1 brood chamber, hoping that you have brood in this box, right? Otherwise, you don't have to use it. So 3 gram, 4 gram, and then 3 applications, 1 week apart. You may argue, can you do 4 days? Probably you can do. But this was following the regular norm that other people have done, 1 week apart. We looked at all those different things, mite infestation, colony evaluations, looking at any stress on the bees, doing some physiology like worker oxidative stress. We looked at queen health and all. I'm not going to cover everything today, but so this is how we do. I think Fonta was saying you have to cover your hive really well. That's really important. I think Ken O'Grain probably has done a lot of knowledge here. He has actually, I borrowed some of his ideas here, putting the shop towels in the front, or not shop towels, wet towels in the front, so that you cover the hive as much as, and you see all those blue tapes and all, we close all auger holes, because that's important. If you have a lot of openings, then that's not going to give you good efficacy of the product. So you have to close them well and at least get the vapors for 10 minutes for sure. So we did like a 11 minute, I think, something on these hives. So we use that vaporizer. I know people use like all those bazooka type ones, but we wanted to test only this one that is more popular with backyard beekeepers, the Varox one that you're seeing there. So again, quickly I'll show you some data because of time. So you can see first 24 hours, right? One day you see the drops. In that one you see about 1400 plus mites drop. In the 4 gram. 4 gram is the top one, the dark. The green is your 3 gram dose, and this is your control. So control also, we still put the wand in. So experiments are done like that, right? Control doesn't mean that you don't do anything to the hive. Even that you have to disturb the same way. Otherwise you can't publish a paper if you just say, I did control means I didn't do nothing. So you still put a wand, and with empty, no oxalic acid, you still put it in for 10 minutes and heat it up. So that's what your control is there. So you can see there is still some drop after at the end of second day as well, but it's not as much, right? So we still don't know. Maybe it recrystallizes on the bees as well to some degree, and it's still giving you some efficacy. So after that, you don't see much. Like on day 3, like you see, it's all the same. Controls are almost same with the 4 gram and 3 gram. So this is after the 3rd application also. So for new beekeepers here, don't just imagine one time you put oxalic acid and your mites are gone. No, right? Because there will be mites coming every day out of those cells. Because if you suppose your queen lays 2000 eggs in a day, so there are 2000 larvae, just I'm making some numbers up here. So there is 2000 larvae every day, and if half of them are infested, so there is 1000 mites already there in the cells. So 1000 mites might be hatching every day as well. Again, I'm not scaring you, I'm just making up some numbers. So think about so when you use the oxalic, you are killing only the mites. I think that was Fanta was saying something when I arrived. When you're using oxalic, you're killing only the mites that are on the bees, the phoretic phase bees, the dispersal phase bees, mites, not the ones that are sitting inside those cells that are still in the reproductive phase. Because oxalic does not penetrate the wax comb or the cell. Only formic does. If you use formic acid, I think formic does a good job penetrating the cells and going back and then going inside and killing the male mites and females as well to some degree. So with this one, that's what you're seeing. Like after 3rd application, I'm still getting more mites, right? I'm dropping like 2000. So if I thought all mites are gone, you shouldn't have any mites left during my third application, right? So I'm still getting mites. So you have to repeatedly use the oxalic. It's not like you can just do one time and say all mites are gone. It's not the case. Okay. So we looked at some larval mortality as well, and so only 4 grams. I'll be really quick on this one. So you see that 4 gram is the blue one there. So we are seeing some mortality there with 4 grams. So 3 gram is not statistically different than the control. So that's what we are telling people, that 3 gram appears to be the most effective dose because it's not only working well like 4 grams, but also not killing a bunch of larvae there. So I think 3 grams probably is still a good dose. Again, we submitted this for publication, but you don't have to rely on researchers like us, because EPA is the one that changes the label. Hopefully when studies like ours and other universities are also doing similar things, so hopefully the label might change once they realize that 1 gram doesn't work, and maybe then you can, but still the label dose is still 1 gram, right, as of today. So I'm not going to change the label. Okay, I'll skip this nutrition. We do a lot of nutritional studies as well, but this was a big study where we wanted to create a database for bees in terms of pollen. So we collected about 100 different pollens. So a lot of volunteers helped us collect pollen. So we are looking at, so let me just see. If I go 5 minutes over, it's okay? Yes. Okay. Let me see. I think I have 1 minute left, so I'll wind up in 5 minutes. So this is the lame example I'm giving you, right? So you probably have heard people ask you, oh, give me top 10 plants I can plant in my backyard, right? Or probably you have asked someone like me, give 10 plants that I can put for my bees, right? It's a great question. So how do you tell them 10 plants? You just have read somewhere, or you have some experience, oh, lavender, very attractive, I see a lot of bees, so let me say lavender, or maybe something like bachelor's button or something you randomly give, or you have some experience, right? So again, I'll not, in 5 minutes I can't get all your inputs, so I'm going to tell what I want to tell. So the most of the plants today are selected based on attractiveness. Probably most of you agree. Attractiveness in the sense, I have a row of lavender here, a plot of bachelor's button, phacelia, or something attractive, a bee balm, or whatever you think of, and then you count bees, how many are there. And you say, oh, I'm getting 100 bees in a minute on this one and 200 bees on this one. So based on that, you say, oh, this is more attractive to bees. And so what I'm arguing here is, does attractiveness always mean good to your bees? So that's what my lame example of French fries here is. If I put both together, what would you eat? I would first get some French fries. And someone in the audience joked last time, they said, yeah, we would eat French fries first because they'll get cold. So first you have to eat French fries, then salad. But the reason I'm giving you this example is, attractiveness doesn't always equate to nutrition, right? I mean, French fries are very attractive, but are they same in terms of salad to your nutrition? Probably you'll get some good carbs, but probably not other things. So same with bees. So the whole goal here was to come up with, I'm not saying that these things are bad, right? If you see this database, plant database from USDA or Xerces Society in Portland, these are all very good lists of plants that you can plant. But the whole goal is to add another column and say, we collect pollen and say protein content, amino acids, sterols, omega-3 and 6 fatty acids. If I give you all the information in that column, wouldn't that be a good way for you to go and say, okay, I'm going to choose Phacelia, but let me look at the crude protein, amino acids, and that would be a more scientific way to choose plants, right? Not just randomly say, oh, this is attractive because bees are coming. Bees will come if nothing is there, right? They will come to anything if it's attractive and nothing else is. So if you give a choice, then probably they'll do something different. So that was the whole goal of this another USDA study that we got funded a few years ago. So we designed a pollen vacuum because pollen collection is a painful task. If you have done that before, it's a manual collection of 1 gram. So one of the students designed this pollen vacuum. I still have it in the lab if someone wants to try more pollen in the Eugene area in the spring. We trained a lot of citizen scientists to collect pollen for us as well. Maybe someone in your group also has sent us some pollen from different plant species we want. So this is what we are trying to see with all the sterol profile. So this will all be ready in the next year or so, and it will go to that plant database and people can use it. Any conservation group that wants to plant plants for bees, then they will have a more scientific way to choose plants rather than just randomly looking at the attractiveness. Okay, I'll skip this one, protein study, and quickly go into tropile labs and finish this. So just be mindful of this. Again, don't get worried, because tropile labs, probably we have been worrying about this for 25 years now. It hasn't arrived, but the only reason why they are worried now is because of this. You see that map there? This is where all the red, all those countries you see, India, Nepal, China, Thailand, Vietnam, South Korea as well added now in the red. So South Korea didn't have this mite 3 years ago, but now they are seeing that mite there. And people were thinking, because South Korea is not as tropical, they have still broodless periods in their country. So people thought maybe this mite will not be successful there, but they are seeing in South Korea as well now. So this has created a little more urgency on this new mite, and people are worried a little bit more. So I'm not like totally concerned, but I want you all to be more vigilant, and I'll show you how to look for this mite. It's not that easy task. It's a very small one. It's only 1/3 the size of your varroa mite, which is already a disaster. So this created the news. That's why you are hearing more about this, is because it's spreading. And now they have seen, if you see that Georgia, not the Georgia in the US, but the Georgia, the country, and then there is Uzbekistan there, and then Ukraine is somewhere close by there, right? So Ukraine is not still listed in red there, but they are thinking they already got the mite as well. So that's why and Canadians, they import bees from Ukraine, the package bees, I guess. So there is a scope of getting package bees, and if there are a couple of female mites they end up, then you're done, right? So that's how varroa mite came into the United States. Maybe we still don't know the route. First it was discovered in Wisconsin, but people think it came through Texas. Maybe there was a ship that had a swarm hanging from some of those countries where the mites were, and it just swarmed onto the inland, and we got the mites from those the mites that came with the swarm. So again, we don't know the route, but this is why it's concerning. And so these mites initially were in Apis dorsata. Have you heard of Apis dorsata? It's a huge single comb. You see that one here on the left here? You don't see those bees in the United States. Here, the new beekeepers here, we have bees that are cavity nesters. They will be in the box or in a tree hole or somewhere else. They will not nest like one big comb on the branch of a tree. This only happens in the tropics where the weather doesn't get too cold. Otherwise, those bees will die. So this mite usually was there probably 70, 80 years ago. They found on those. But now those mites have migrated into your regular Apis mellifera, which is the European honeybee as well. So I think it just has switched her host. So they are moving into different species of bees as well now. So that's another concern. Okay, so again, I'll not go into too much of the life cycle here, but I'll just give you why we are concerned is faster reproduction. So they can reproduce faster. So what do you mean by reproduction faster? But they are still in the brood, right, inside the cells, like your varroa mite. The biology is almost similar to your varroa, but there are few differences. So I have put the differences on the right side here. So their faster reproduction means they have to come out along with the bee, right? So the reproduction time is the same, right? The inside the cell in the brood area, in the brood, they are staying the same. But can you think of how the faster reproduction, what does that mean here then?
More of them coming out.
Yeah, that could be one. Yeah. And again, yeah, quicker
Population growth.
Yeah, so that's what I'm saying. Quicker population is possible only if they can get out of those cells. They are capped in those cells, right? They are not getting out. They have to come out only when the bee comes out of that cell. Think about one more option.
Adaption.
What is that?
Adapting.
Yeah. Yeah, we are getting close, but still I'm looking for the exact answer here. So
More options for viruses to.
yeah, more viruses and all, yeah, that's the thing, but I'm talking only about, think about just the reproduction part I'm asking. So what they are doing is, you all know that varroa might stay on the bee as well for a week or so, and then they go into the cells for reproducing, right? That's what you probably heard from varroa biology is, varroa doesn't come, like suppose there is a female that comes out with one baby, she doesn't immediately find another cell to go in. She sticks on to some younger bees, nurse bees for a while, maybe a week or 10 days, and then her physiology changes, she recuperates her reserves, and then she goes to the next cell next time, right? That's what we all heard. But this notorious one, it doesn't wait that long. It comes out of this cell, goes into a new cell immediately. So that's what I mean faster reproduction. So that's one way. But they can't reproduce anything inside is the same. Maybe as you said, they may produce a little more babies, maybe 2 maybe. So again, the biology is not very well understood still. You laugh when I say, oh, we don't, because in those countries, these mites have been there for a while, and those countries don't have enough money to really spend on studying those type of mites. So there is still a knowledge gap in terms of biology. So that's the reason people from US are going and studying there as well, like the Auburn group, or maybe in the future if it gets more, maybe we will try to go and study there as well. So we don't know at this point. Again, so yeah, that's the difference between those. So it looks like this, you see, and the other big difference is you see all those dots on the larva there. So they have punctured the larva that many times, whereas your varroa mite only punctures once, and she feeds only from that one hole. But these guys will do multiple holes onto the larva as well. But of course they are a little smaller, so the holes might be a little smaller. So that's another problem with these guys too. So that's how they look like. So here is the Tropilelaps is the one here on the left here on the lower front. Those are all look-alikes, so you may get confused. This is your varroa mite that we have, but there might be some flies like Brawler fly or some pollen mites that might be confusing as well. So I got one vial here, and unfortunately the lighting isn't great here, so I may not show you today, but if you want to come see, then come to the lab, but at your convenience, and I can show you under the dissecting scope would be better, but they are visible to your eye, but very small. So that's how they should look like. If your varroa mite is that size, on the left is your Tropilelaps, which is 1/3, right? If you put 3 of those on top of varroa, they will fit there. That's what they're showing on a penny as well. So that's how they look like. They're more longer, not like your varroa is more wider, right, like this, but they are more longer and little ones. So yeah, keep an eye, and this is the last slide, I'm done. So they call a bump method, the one you see in the dark highlighted one there. The bump test is basically you take a frame and you have a Tupperware, a white background or something, and you just bump it. I mean, literally all the larva, everything has to fall. Because these mites are so quick, and you can't, and why can't you do an alcohol wash? You all do alcohol wash or sugar shake to find your mites, right? Why is not a recommended method? It's there in the list, but it's not a very efficient way to find. Why?
Well, because they don't spend much time in their frets.
Yeah, absolutely. So they are not on the, they don't like to be on the bees. They are always either stealing, getting into the brood and reproducing. And I'll show a video and then I'll stop. I have a neat video where you can see they're running around on the hive, on the comb. So that's why people think alcohol wash or sugar shake is not a great way to look for these mites, because they're always running around and they're on the comb. So if you bump the frame, hit it hard on the surface, then you'll see them fall and you can identify them that way. Can you see the mite on that?
Yeah.
That's where she is, yeah.
Wow. Oh my God.
I think this is a video probably. No. You see them crazily running like ants? Your varroa doesn't do that, right? You hardly see them running like that. They are either on the bees or they are in the brood, or sometimes you'll see 1 or 2 here and there, but you see literally they are like ants or something crawling around. They go very fast as well. Show you one last one and then we'll stop. You see them running around there? Okay, I think I'll stop and take questions if you have. Thank you very much. Yeah, Vanda.
I have 2 questions. One is, I'll first start with the Tropilelaps. Is any of our treatments been shown to be effective on it, like oxalic acid or formic acid?
Yeah, good question. So For this one, they think at least, I mean, as I said, there's not much research yet, but they say making it broodless and use formic, that's the only way you can get rid of those. Because it kills inside the cells, right? Formic is the only one. Because your oxalic may not be effective because they are not on the bees. So you have to use formic, which can penetrate the cells and kill those mites. And if you can get a brood break, and one important thing I missed is these mites, people think in those countries that without brood they can't survive too long because they can't survive, they don't feed on adult bees. That's what the assumption is. Maybe that will be wrong if you start doing more research. But they assume that they don't feed on the adult bees. So if there is no brood, like when we don't have any brood in December or January, like for our temperate weather, maybe they will not be, that I think one of you said, because it's cold here, so those mites may not survive. You may be right, but who knows? If there is even a little brood, that's enough for them to survive. And in December also, sometimes, some years, we see a small amount of brood. So we are a part of a big study from Auburn and other universities where we are monitoring brood for the last 5 years. So we have 10 hives designated for brood monitoring. And our students diligently go every 2 weeks, even if it's raining or cold, whatever. They really open the hive quickly and look for the brood area to see if the queen still is laying. So this year we didn't see any stop in brood. There was still a small area in December as well. So if you end up in that scenario, these mites can survive in that brood and be there for, yeah. And what was the next question?
It was about if you could speak to the resistance that the like amitraz and different things, treatments that are resistant. A lot of people kind of have this idea that it just makes you resistant, the bees resistant to that particular treatment. But I read an article recently that said when they become treatment resistant, they're resistant to all treatment, not just those ones that cause the resistance.
So you're talking about varroa, right? Not the bees.
Varroa.
Yeah, okay. So yeah, that's a good point. So there is something called cross resistance. That's what she's talking about. As researchers, we use the term cross resistance. Means if it gets resistance to amitraz, then any other compound like your oxalics, formics, none of them should work either. No, that's not the case with varroa. We have not seen any cross resistance with varroa. So it's only amitraz that will, even with amitraz, I'm still seeing some efficacy. I'm not saying it's totally resistance yet, but maybe in 5-10 years I may see a resistance problem. But that doesn't mean that other products don't work. No, the other products should work because the mechanism is very different. I know I don't have another hour to explain how resistance works. I can happily be here until midnight, but so these are all mutations, right? I mean, there is a selection pressure of one miticide, then 3 or 4 survive, then it propagates that genetics, right? With mutations, there is a cuticle that they can increase in their density of the cuticle, or it could be, we call cuticular penetration resistance. There is metabolic resistance. Sometimes they can ingest the compound and degrade it inside as well. So there are different ways of mutations that happen to create resistance. So that's why each compound is very different. So if they are resistant to amitraz, doesn't mean that other products will stop working. They will work because their mode of action is different.
Okay, great. Thank you for
Yeah. Other questions? Yeah.
So when you said 3 grams of oxalic per brood chamber, are you referencing like westerns or deeps? Does it matter? Kind of the size of the chamber?
That's an excellent question. Actually, I don't think I have a good answer. But you see, all these companies, when they make, if I was the company making this product and giving recommendations, I know there's no company, right, for oxalic. USDA is doing a favor by getting the registration, and they are, otherwise no one was even willing, because they can't make any money selling oxalic acid. Because you can go to a hardware store and buy your own oxalic and try. That's what people were doing in the past. Now you have that, what is the word? Bioxol, Apis Bioxol, what is that company name called? Yeah, AP Bioxol or something. So they are just a namesake. I don't think they're making money. USDA gave them the registration, and so that's how the product is. We don't need to get in there. But I'm saying, like, most of these companies, when they do research, they don't do real good with different dimensions. And so, yeah, it's a very, very good question, actually. You can argue, are we talking about, so I think they will argue, they'll say just the standard Langstroth hive that you have, the dimensions, the standard ones, not Westerns or others. So they will say that's our recommendation. But I still am doubtful if they have any math done for really getting how many bees are there covered. Yeah, I mean, if you're a scientist, you will go into...
Were your studies
So this was all Langstroth. The one I was showing all the studies is the regular. So most of our research will happen with the most popular design that beekeepers use. So commercial beekeepers will be always a standard Langstroth type, right? So we use those for that study. But a great question.
Probably deep boxes.
Yeah,
Deeps like-
yeah, deeps. If you want to use, I know I'm bad with beekeeper terminology. As a researcher, I should brush up more beekeeper terminology, but sometimes I don't use that very well. So I apologize if I confused you. Yeah. Any other questions? Okay, it looks like that was too much for you for today. So take rest, sleep well, and if you have some pressing questions that are waking you up in the night, then email me or call me. I can answer those. Thank you.
So, Dr. Sagili, thank you very much. Appreciate it.